Well, thank you very much, Karen and Xiaochen, and as well as all the other organizers for giving us a chance to tell you today about some of the dynamic properties that we try to engineer into microenvironments. And there's a very important reason for why you would want to have a dynamic system to signal to your cells. So although Xiaochen had mostly talked about three-dimensional materials, as Laura did, and our materials can be used in 3 dimensions as well, I'll primarily tell you about some 2-dimensional sort of proof of concept studies that we've done to emphasize the importance of dynamics in your system. And so I'd also then like to start out by thanking those that did the work. I tried to highlight or put in bold all those, including the first story, Jessica Nguyen and a former Ph.D. student from the lab, Ludovic Vincent, have a poster today, poster number 4, and I'll tell you a brief vignette from what they've presented with a great collaboration with Xiaochen, as well as then some work that I'll present later, mostly from Jennifer Young, and then Matthew Ondek and Aditya Kumar, as well as then I'd like to thank our funding sources, primarily NHLBI and the Office of the Director at NIH. So suffice it to say, we've spent the past hour or so talking about this extracellular matrix, and I won't belabor the point that this is a 3-dimensional fibrillar structure in which cells reside. And as was mentioned earlier, they have very important intrinsic properties. So whether we're talking about the composition, the topography or sort of scaffold structure, the porosity that can dictate things like cell shape, as well as then what I'll tell you about, which is matrix stiffness or elasticity. The appropriate term would be Young's modulus, but we'll just call it stiffness for right now. And that's the concept that cells can live in very dramatically different environments. So they can be in something that's relatively hard, like this, for calcified bone, and something that is way too soft as well. So that's my one joke. All right, so the way that you can get at this intrinsic property of Young's modulus, or what I'll refer to then as stiffness, would be an idea of like a bowl of spaghetti. So you can have your pasta noodles, and you can have a low amount of noodles, or you can have a high amount of noodles, and that effectively will make things stiffer. The more bulk you've got, the stiffer that your material can be. But you can also cross-link it together, and so I like to think of that as sort of half-cooked pasta, where it sort of sticks together at some points, and so as you pull a network that looks like that, all the other noodles then translate with the noodle that you've grabbed onto, and so you effectively can make the environment stiffer, either then through having more bulk in terms of entanglements then, or more crosslinks. And so the idea then that we first started out with was whether or not stem cells could respond to these sorts of cues, and as we've shown just about a decade ago, that irrespective of the specific growth factors that are in your culture, if you make a material soft or stiff, cells can intrinsically respond to those types of properties. And this has not been shown just by our group, but by a variety of others, from Helen Blount's lab, David Mooney's lab, and so we've looked at a variety of different stem cell types that seem to respond to this, so not just adult stem cells, but embryonic, iPS, tissue-specific stem cells like Helen Blount's lab works on, and I believe Tom Randa will talk to us about later today. And so you can also do these in 3D, as Dave Mooney's lab has shown. But really what's, the field has sort of come to a crossroads in the past couple of years, and that was put forward by Fiona Watt's lab in England. And what she said was perhaps this idea of stiffness, and I'll get to the dynamic part in a second, but maybe stiffness can be modulated by the microenvironments ligands and how you have cross-linked those ligands to your material. And so they brought up this idea of matrix tethering. And like I said, we just recently published this in Nature Materials, so, and there's a poster outside from Jessica and Ludovic. But the take-home message is that when we look at tethering, and we measured it, and we think that it's real by AFM, but if we looked using a fret probe to see whether or not the cells were actually tugging on the probe itself, we didn't see any difference in deformation as a function of stiffness or tethering than this sort of first column then on the plot. What we did actually see was that the cells could pull on their microenvironment in dramatically different ways. So if it's relatively soft, they pull a lot. If it's relatively stiff, they don't pull so much. But the amount of force that they're using sort of scales with this, and the important take-home message is that if you think of this as sort of a linear series of springs that the cells are pulling on. What really matters then is the spring that deforms the most, and that's this hydrogel spring relative to any sort of matrix tether. So if you were to look independent of matrix composition, the stiffness again dictates. So if it's soft, cells become, adult stem cells like a mesenchymal stem cell become fat-like. If they're stiff, they become bone-like. And so this is alkaline phosphatase then and oil red O. But what I really want to get to then is this idea, if it's not so much the tethering, what sort of dynamic changes occur in the microenvironment that could affect then something like stiffening, and you can get spatial changes, or you could also then see temporal changes. And I want to focus in on the temporal today, because I think that that's a more interesting story, and we've got a lot of data from that. So several years ago, Jennifer Young, a graduate student in my lab, was really interested in this process of differentiation and ultimately then specification of the heart itself. So moving from a contractile heart tube, ultimately going through a U-shape and an S-shape looping pattern into then a four-chambered heart. And what's great about development is that there are a variety of cardiac models out there, whether you're talking about mouse, rat, or in our case then, this is an avian system. And so we could pull out the myocardium at various time points, either starting out at precardiac mesoderm and moving all the way through a fully four-chambered and mature heart. And what we saw was that there was this interesting increase in stiffness. And that is correlated then with an increase in the expression of matrix proteins. So we thought, is there a way to modulate this or to mimic this dynamic process in vitro and sort of in a reductionist way, probe to what extent does this temporal change also help program stem cells as they're differentiating into mature cardiomyocytes. So we could use our favorite material for this process, which is hyaluronic acid. We couple a free thiol group to that, and this is one of two chemistry slides, so I'll breeze right through it and just say that we form this cross-linked hyaluronic acid material that you can see moving from left to right increases its cross-linking with time. So very large pores here, smaller pores there, lots more cross-linking. And we can tune that dynamics simply by changing the molecular weight of our spacing arm, the cross-linking arm, or by changing the amount of sort of bulk polymer in your system. So those are two ways that you can change either the starting modulus, or stiffness, if you will, for the material by changing the bulk properties, or by changing then the molecular weight of that spacing arm, which tunes the dynamics. So the longer the arm, the faster the cross-linking. And this is going to be a process that's ongoing in your culture due to a Michael-type addition reaction. So the way that we can quantify the maturation of these cells is to look at the spacing then of their sarcomeres. And this was an assay that was developed back in the 90s with Jean and Joe Sanger at UPenn, where you simply look at the spacing and typically mature striations, if you're not too familiar with that, depending on the type of muscle, tends to be about 2 or so microns. So this repeating unit that's contractile repeats itself every 2 or so microns when it's mature. Immature versions of that then have shorter spacing, and so if we were to look at animal or cells that we had pulled out of the chick in a very late developmental stage, then all the cells look mature. So they have striation spacings that tend to be above 1.8 microns. If you go earlier and earlier, though, and keep the cells, look at the cells at the same total age, so if we pull out the cells at 72 hours post-fertilization, we then look, say, a week and a half in vitro later, and assay whether or not the cells on the right in the static material or in the dynamic material on the left have matured. And there's a striking difference then. The cells on the dynamic material, 80 or 90% of them tend to have mature striations, and that's not necessarily the case then in the static environment. Now, of course, the cells are responding to a variety of growth factors that are contained within the serum that we also provide these cells, but absent any specific growth factors, the cells are simply responding then to this dynamic environment, we think. So the question came up of what is this response and how are they responding? So there are a variety of different literature reviews that have talked about how cells respond to these sorts of mechanical insults, if you will. And so our favorite signaling pathway came about from a very large protein kinase screen that we did in collaboration with a company called Kinexus in Vancouver. And so we were able to tease out from about 850 or so protein kinases, a subset of kinases that tended to be upregulated as a function of both time and being in this dynamic environment. And so we were interested in drilling down with those proteins. We could also then figure out their ontology. So what sort of signaling pathway are they involved in? And when we did that and segregated it by agonist or antagonistic pathways, we saw a inverse correlation in a dynamic environment, meaning that the antagonists are going down, the agonists go up, and that's not necessarily what you see in a static environment. So with the help of Alex Zambon, a bioinformaticist at UCSD, we were able to come up with signaling maps, and what I'd like to highlight then is just this one map here, where you can see the AKTs are highlighted in red on one side and green on the other, just an indication that in the dynamic material on the left-hand side, they are being upregulated or phosphorylated, and on the right-hand side, in the static material, they're being downregulated. And so if you're a cardiologist, you'd be familiar with, or a developmental biologist, you're probably familiar with the fact that AKT and inhibiting GSK3 beta is a classic pathway that's associated with the development of cardiomyocytes. But again, the interesting thing here was that we weren't specifically activating either of those pathways other than just providing basic serum proteins and providing this dynamic environment. So we came back with a set of inhibitors, and the most interesting story then is if you inhibit AKT, you block the effects of the dynamic material, whereas if you then inhibit the GSK3 beta in a static environment now, you can upregulate that, GSK3 beta being an agonist then against this differentiation pathway. So that was really intriguing to us and hinted that perhaps the dynamics really matter and they can act through very interesting signaling pathways that were previously just associated with general differentiation. But this, again, would be mechanically induced. So the other brief vignette that I want to provide you in the last 3 or 4 minutes then is another type of hyaluronic acid material. So this, again, is HA. Instead of putting a thiol here, we put a methacrylate. What this enables us to do is either using DTT or UV-activated cross-linking, we can then get a dynamic environment. So you can go, and this is an on-demand cross-linking rather than a gradual one. So you just simply add in your UV and your initiator, and the material will cross-link. So we can go from something that's soft and relatively myocardial-like to something that's much more like a scar that you might see in a myocardial infarction, simply then by this UV cross-linking step. So let's see if this movie will play. Everyone that works on cardiomyocytes, like Xiaochen just showed, has to show a movie of their favorite beating cardiomyocytes, so we can make these cardiomyocytes. We start out actually from a very interesting population. This is a collaboration with Eric Topol and Kristen Baldwin at TSRI. So the cells, you can't really see it, but they start out as positive iPSCs that we pulled out of patients that have an interesting set of risk alleles for in the 9p21 locus, so that predisposes ones to coronary artery disease. And what we've found is this very interesting phenotype. Once we differentiate them to cardiomyocytes, we put them into this bioreactor that we like to call myocardial infarction in a dish. So we induce hypoxia, and then we stiffen the gel, much like one would see in vivo. And interestingly enough, if we can get these movies to play. So in the soft environment, you see all the cells sort of flash together, and this is indicative of mature calcium signaling. If you were to then look in the stiffened environment, so this started out soft, we induced hypoxia and then made it stiff, and mimicking sort of the time sequence that you would see in ventricular remodeling in vivo, and it sort of looks like a Christmas tree. In that, the stiffened cells flicker on and off in an uncoordinated fashion, whereas in the softer environments that stay soft forever, then you see this very nice regular pattern of beating. So those are a few examples then that I've been able to provide you where we think that the temporal dynamics are increasingly important in terms of differentiating these cells, so they can improve commitment, or in a specific example then perhaps model a disease process. So improve development or maybe harm the commitment then of these cells. And also then that can be regulated with specific sort of mechanosensitive signaling pathways. And just as one last point I want to make it's very imperative that you consider the translatability of these materials, and so everything that I've told you today with dynamic environments really would have trouble then being translated in vivo. And this is just one example that I want to close with. So this is our original material of thiolated HA, and this was actually work with Karen, where we took the material, everything that we had done subcutaneously would have suggested that this material was biocompatible, but we found huge granulomas that resulted once we injected this material into SD rats and waited a month. So we took this to our immunologist friends, and they said, oh yeah, this looks like a classic granuloma, and when we looked back in the literature, we found, oh, the mechanism that we were using, if I could just flip back to that one slide with the chemistry, having free files is a really bad idea in highly vascularized situations because macrophages love to reduce free files. So essentially what we were doing was creating a nice environment to home various parts of the immune system to this material, and so that was very unfortunate for us. So that's my one caveat, and I'll stop there and turn things back over to Karen.